I'll open up my presentation with this slide and ask you to look at your left, look at your right. What do you see? Everybody's different, right? We have this wonderful diversity of people all over the world. And now take a look at this slide. This is our most common research model. These are inbred animals, inbred rodents. They're the most common in biosciences research model. And if you just take a look at these pictures, you can realize it's really difficult to capture human diversity using the most inbred animal model because we are the least inbred on the planet. We consciously avoid inbreeding. But over the past 10, 15 years, perhaps there is a solution to this problem. Perhaps there is a way to create organs in a chip and microphysiological systems that together with patient cells or iPS cells could help us model disease, human disease, in a very diverse population. And so I usually argue that organs on a chip arose from tissue engineering because we use a lot of the same methods and principles. If you look here in tissue engineering, there was an idea that we could take cells from a patient, put them on biomaterial scaffolds, expand them, grow the tissue in a bioreactor, and then put that back into the patient. And when I was an undergraduate student at McMaster in the late 90s, when I read this in a paper published by Bob Langer, it was in Scientific American, I was completely mesmerized. I said, I'm going to dedicate my life to tissue engineering. But there hasn't really been as many successes in clinical translation as we thought. The main reason for that is really hard to keep large pieces of tissue or organs alive because we can't solve the vascularization issue. And everything inside a big piece of tissue becomes dead during tissue cultivation or upon transplantation. And then through organ on a chip engineering, we just decided to make really small tissues and organs. There is no whole organ on a chip. It's usually just a tissue that we are capturing. When we decrease the scale, now we can keep the entire tissue alive most of the time. And instead of using these tissues for repair, we use them to study mechanisms of disease and to develop new therapeutics that could help the patient. But here is another indirect proof that organ on a chip arose from tissue engineering and microfluidics. You can see the number of papers published each year in the area of tissue engineering, starting from the late 80s and 90s when this field was really defined. You can see exponential growth of the field. And at the same time, George Whiteside figured out how to make microfluidic devices from silicon, PDMS. It's silicon with an E at the end. And how to put cells in these microfluidic channels. So together, using advances from these two fields, we can see that organ on a chip arose. And really, Don Ingber used that word the first time in his science paper in 2010. But people were doing this kind of work even before. And you can see the field is really on an exponential increase. And now if we think about where does organ on a chip fit? And obviously, the structures that we are making, they're larger than protein and cells, but they're nowhere nearly close to the complexity or the size of real organs. So we operate somewhere between micrometer scale to at most millimeter scale. And these tissues that we develop, the unique things about organs on a chip is that we can measure function. You know, people who grow organoids, they operate on the same scale. But one important distinction between organs on a chip and MPSs and organoids is that we really focus on measuring function. If we have a heart tissue, we want to measure contractility. If we have lung, we want to measure permeability. And sometimes it's difficult to get those functional readouts from organoids alone. And really, we can use these systems then to model disease, develop new therapeutics, and study drug responses. So one of the overarching hypotheses of the whole field, because it's really bioengineers who focused on developing this field, is that structure will enable function. That's one of the cores of engineering approach, right? There are pillars in this building, posts that are holding the whole structure. And for us, if we want to make a cardiac tissue, we are focused on taking cardiomyocytes and aligning them in parallel. This here is an AI-generated image. The myofiber alignment in the heart doesn't really look like that. It's supposed to be here to capture your imagination. But we want to take individual cells, make sure they're elongated, they're aligned in parallel, and that when they contract in unison, so we can measure the shortening and the force of contraction. But that's just one type of structure. If we take a different look at the heart, we take a look at the vasculature, we see something completely different. Now we see fractal structure. These are not aligned vessels, but they're branching from the largest to the smallest one. And this is actually a real cast of vasculature from a mouse heart. So you can see the vasculature is so dense that it gives the shape to the heart. So over the years, my lab has worked in both organ-on-chip engineering and instructive biomaterials. We had the translational efforts in both of these areas in organ-on-chip by creation of this company called Tara Biosystems that commercialized our organ-on-a-chip, heart-on-a-chip, and also some of the hydrogels that are being commercialized through Qtera. But today I'm going to mostly talk to you about organ-on-a-chip, our BioWire, AngioChip, AngioTube platforms. So one of the key ingredients of our approach is this elastomeric polymer. It's called POMAC. It's a copolymer of 1,8-octanediol, maleic anhydride, and citric acid. This polymer is a viscous liquid. It's very easy to make because if you talk to a chemist, I'm a chemical engineer by training. If you talk to a chemist, they say we are not true chemists, which is probably true. So we can only do simple chemistry, but we are not true biologists either. So we are kind of in between, right? And it's very easy to make this polymer. You can just take monomers, put them in one pot. You're going to get, if you elevate the temperature, you melt everything, you're going to get this linear prepolymer. And this linear polymer is viscous. And you can mold it, you can make it flow through PDMS stamps. Or you can ink the stamps into prepolymer, and by UV illumination, you can cross-link the polymer and get the shape that you like. So not only that you get the shape, but this elastomer is elastic, and we can tune mechanical properties to make it fit mechanical properties of the heart tissue. For example, if you want to work on the heart, that means that Young's modulus will be around 200 kilopascals or so. We introduce porosity by using a porogen, PEG, and when we wash out after cross-linking this PEG porogen, you get these nanopores, which means that the polymer structure will be permeable. It's going to have a certain predefined shape. It's going to be elastic, mechanically suitable for cardiac cell culture, and it's going to be permeable. So using this approach, we made many devices. And Yimu Zhao from my lab, she published on this BioWire 2 platform in Cell, where she figured out how to grow this string of cardiac muscle by just growing cells suspended on 2 polymer wires. And so she would take basically a piece of plastic, tissue culture plastic, use hot embossing in a really nice craft facility at the University of Toronto, create this microwell. Into the microwell, we would put polymer wires, polymer that I talked about. We would seed the cells, iPS-derived or human embryonic stem cell-derived cardiomyocytes into microwell. And then we would apply electrical stimulation using these electrodes. And that's how we force the cells to align, and we create a piece of cardiac muscle that can beat. And this is what a piece of cardiac muscle looks like. It's just a string, it's beating. But if you take a look under blue light, basically, you look under fluorescence. Right here in the middle, you can see that the wire is deflecting, it's moving, which means that if I have calibration curves, force displacement calibration curves, I can figure out force of contraction. I can figure out the functional property of this tissue just by looking at it. And we've grown these tissues up to 8 months, and these wires remain blue because this is autofluorescence of the polymer. It's not an indicator or a dye, just the bulk of the polymer is just autofluorescent. And then we would apply electrical stimulation. And this is something we actually were doing even earlier. We had a paper in Nature Methods back in 2013 with this principle that basically we want to stimulate and make these cells beat as fast as they can, like push the tissue to the limit of its capacity. It's like interval training. If you push yourself to the limit of your capacity, you're gonna achieve results faster. So that's the same idea here. We would increase the stimulation rate up to 6 hertz over 6 weeks. And at the end of this period, we get a tissue that has this nice positive force frequency relationship. If we turn off the stimulator, the tissue is quiescent. At least for about a day, it will be quiescent. And then if we apply one slow stimulus, we see this increase in the force, which is called post-stress potentiation. And that occurs because there is accumulation of calcium in the cytoplasm. And then after that, when we turn off the stimulator, basically the force goes down. And when we get this mature tissue, it means we have to test drugs under stimulation. And we can see a number of expected drug responses. An interesting one is this E4031. That's a hERG channel blocker. Rodents have this channel, but they don't rely on it as much as humans. Humans really rely on it for when they want to return the action potential back to the baseline. And you can see the drug that blocks this hERG channel causes very, very dangerous rhythm disturbance. And that has actually happened with some drugs post-approval, such as Vioxx. And Vioxx, for example, killed about 27,000 people in the United States alone. And this shows you that the platform like this is capable of picking up effects of known drugs. When we worked with iPS cells from various patients, we were able, using this platform, to capture phenotypes of various diseases. And if you look at genetic disease, using cells that actually have mutations in myosin heavy chain, and these are from sick kids, we were able to capture that specific phenotype. When we worked with colleagues from Toronto General, we got cells from patients who have mutations in sodium channel 5A. Again, we were able to capture the phenotype, especially after maturation. And as well as when we got cells from patients who were involved in NIH hypergen study, we were able to capture features of polygenic disease, which was left ventricular hypertrophy as a result of high blood pressure. We can also model acquired disease with this approach. For example, we were able to capture features of fibrotic myocardium, as well as features of cardiomyopathy resulting from drug application, such as angiotensin II. And so this technology has been commercialized through Tata Biosystems, as I mentioned. But one of the questions that people always ask in MPS and organ on a chip field, there have been dozens and dozens in just a short time, less than 10 years. There have been probably 70 companies created in this field. And everybody is asking, well, how do they really add value? Who made a lot of money, right? Organ on a chip companies and MPS companies, we don't really have our own compounds, right? We have biology, but we don't have compounds. We don't have our own drugs. And AI companies, they have their own drugs, but they don't have good human biology. And in this case, Tata was acquired in 2022 by an AI company, Valo, that had a lot of AI-driven chemistry, a lot of potential compounds. And together, then they were able to get a big contract with Novo Nordisk, going up to 2.7 billion for discovery of cardiometabolomic drugs. So it's really the marriage of iPS cells, MPS, organ-on-a-chip technology, and AI that enabled creation of significant value. So most of the early organ-on-a-chip prototypes, you'll see that most of the devices are kind of made by hand and operated by a skilled operator. I think in most labs, that's how we still operate. And that a postdoc or PhD student is very skilled, they know how to operate these devices. And there is a really big incentive now to move more towards automation and scaling up production, both device production and operation of the devices. And towards that goal, it's not like we figured everything out, but we are moving more towards 3D printing. And initially, we would have these very fine polymer wires, and we would, manually kind of insert them into the device, which only a few skilled students could do. But then Chinghua Wu from my lab, she used thermoplastic elastomers together with quantum dots. And by using quantum dots of different wavelengths, she can have different colors of the wire. She could have red or blue. And then thermoplastic elastomer, you can melt. Polymeric elastomers, you cannot melt, the ones that I talked about at the beginning. And when you melt them, you can extrude them through a print head nozzle. And then in one step, we create this base that has embedded electrodes. It has wells where we're going to seed the tissue. And then the wires are now 3D printed. So we really scale up the production rate. And also, we don't need a skilled operator anymore. So people also ask me, well, are organs on a chip high throughput? And I say they're not high throughput. And even if you scale up production rate and you automate seeding, you have to wait for the cells to make the tissue. It's just, you cannot overcome that time. The cells just need that time. That's biology. So it's going to take a couple of weeks at least for you to get a tissue that's functional enough so that you can measure something reasonable, that's mature enough. So we are definitely not high throughput in the sense of that word that pharma uses, where they screen 20, 30,000 compounds in a period of just a couple of days. We need time, but we are high content. You can get a lot of readouts from one tissue. And just to show how we can get even more readouts, we were able, through a collaboration, to make these electrodes. They're as thin as basically your hair, and they're made of P.PSS. So they're very thin and flexible, but they're also pretty robust. So you can see a tissue beating here with embedded electrodes. So then from a single tissue, we were able to measure both force of contraction as well as extracellular field potentials. So the field is really moving towards instrumenting these organ-on-a-chip devices some more and having more and more readouts. So that's a really brief overview of engineering cardiac muscle. And in that cardiac muscle that I showed all of this work with summary of these many papers and translational effort, there were basically two main cell types there. There were cardiomyocytes, derived from iPS cells usually, and about 10% of fibroblasts. These fibroblasts, usually primary ventricular human fibroblasts, but they could also be fibroblasts coming out of directed differentiation or CD90 cells. We didn't have any endothelial cells. And as I mentioned previously, they're actually, through single cell, we now know through enumeration that endothelial cells are almost 50% of the cells in the heart. They're so important. And the heart is so vascularized that each cardiomyocyte is sitting right next to capillary. You can see here in two different views, capillaries, and there are cardiomyocytes all around them, right? Cardiomyocytes will actually going to make up 90% of the volume of the heart muscle. And each of them is sitting next to capillary because they work so hard, they need a lot of oxygen. And so it would be very beneficial to try to vascularize this muscle. And over the years, people have tried many, many approaches. They tried to use gene therapy or mRNA delivery. And if you remember, in the early days of gene therapy, in the early 2000s, when people just tried using VEGF, vasculature was not stable enough. Right? And they had to stop those clinical trials. They would just make angiomas. And then different approaches, more and more complexity was added. You have to keep in mind, going all the way to these tissue engineering approaches, if you go from here to here, the more complex the approach, the more likely is going to work if you have a skilled person doing it. But the less likely you will be able to do translation, right? In vivo translation or even translation from one lab to another, because it becomes so complicated that only a few skilled people can do it. So Boyang Zhang now, he used to be a PhD student and a postdoc in my lab, and now he's a professor at the McMaster University in Canada. He said, well, if I take a look at a computer chip and angiogenesis, it's just a very complex network at a micrometer scale. Maybe I can go to clean room and try to make something like angiochip. And here is actual micro CT of angiochip, which is a structure. It's a microfluidic scaffold that's made out of the POMAC polymer, which I mentioned in my opening slide. And what you can see here are conduits. They're channels that are branching in X, Y, and in Z direction. And around these channels can be endothelialized, and then we can grow basically any cell type around these channels. So the way Boyang did this is he made a series of PDMS stamps, and then he would ink the stamp in this viscous liquid pre-polymer and apply the stamp on the glass slide, UV illuminate, and then the polymer would cross-link and it would stick to the glass slide better, and then you peel off PDMS. And if you do that layer by layer, you have to use a mask aligner. So this was all done in the craft facility. You're going to get a structure that looks like this. You're going to see those conduits, channels that are going to be aligned with endothelial cells. And around the channels, there is a lattice, and you can seed cells into that lattice. So please also note that there are microchannels here. And it's actually, this process is manual, but it's also scalable. You can get enough angiochips in a relatively reasonable amount of time in the clean room. But these micro holes that I mentioned, which you can see here as well, they're actually critical because polymers are not permeable. Even elastomeric polymers such as POMAC is not permeable. You know polymers, plastics, plastic bags, Ziploc bags. You put your food in a plastic bag to separate it from the environment, right? And we know blood vessels are not separated. They're actually communicated with the tissue all the time. So Boyang had to incorporate these microholes to enable cells to move in and out of the blood vessels, right? As needed, on demand. And then there are also nanopores from the porogen leaching in the walls. And that enables us to use, to get the proteins to go in and out. So this work now, when Boyang was a student, that was a while back, almost 10 years ago. And again, Boyang is also a chemical engineer, and I'm a chemical engineer. And all we do in undergrad is we learn about pumps, we learn about fluid mechanics. How do you size a pump? How do you select the right type of pump for the process? And Boyang said, we are not going to have anything running on pumps because pumps are really hard to run and operate. It's hard for a biological lab to adopt a pump, right? And really to get used to having all the tubing coming in and out. So Boyang made a bioreactor that looked like a well plate. And each angiochip runs on a unit of 3 wells. Angiochip goes in the middle well here. If you put more media upstream compared to downstream, you will create a pressure head, and this pressure head will drive the flow. And these reservoirs are pretty big that you don't really have to, you're going to have unidirectional flow in angiochip. All you need to do is change media with pipettes, as you normally do. And the first step is to line these vessels with endothelium. We would coat the polymer with gelatin, and then we will line it with endothelial cells. After about 2 or 3 days, we take whichever cell type you want, for example, cardiomyocytes, and you deliver it in a hydrogel around this vasculature, and then you wait for a few days and gel compaction would occur. And what I usually say about this, especially when I write grant proposals, when you're trying to explain to the reviewer what's the big deal about the approach, that the polymer gives you mechanical stability. You can pick this structure up, you can handle it. And the cells like hydrogels, and you can basically use any hydrogel or any ECM that cell likes, right? You know, if you have hydrogels and they're not anchored to anything, everything will just compact into a sphere. So the polymer here gives you that mechanical stability. The nice thing about controlling polymer structure is that I have different ways of controlling mechanical properties. One way is through chemistry. Depending on the ratio of monomers in this elastomeric polymer and how much time I cross-link, we are going to get a certain Young's modulus. But depending on the shape here, you can see that we have the lattice into which the cells are seated. You can see that we made a rectangular structure. It's not a square, it's a rectangle. And depending on the dimensions of this rectangle, we can achieve overall mechanical anisotropy of this microfluidic scaffold. So for example, the real heart, right, is going to be stronger along the fiber direction, CD here, compared to the perpendicular LD direction. And in this scaffold design B, if you look at that mechanical anisotropy, we are mechanically mimicking anisotropy of the adult rat myocardium. So you have a lot of control about what that lattice looks like, and you can match mechanical properties to your tissue of interest. So this is just a picture that shows the endothelial cell staining. After about 48 hours in angiotrip, you can see that it is possible to line these cells and have them proliferate everywhere. We were talking about this earlier. It's actually quite hard to line channels perfectly with endothelial cells, and really the coating is the key. So Boyang used gelatin. You have to figure out which coating to use. And if you use an angiogenic stimulus, and in this particular case, we use thymosin beta-4, which is a peptide that is both angiogenic and cardioprotective. And if you look here, if you apply that peptide around the blood vessels, if you just put it in the hydrogel around the blood vessels, now you can see that the cells, endothelial cells, are trying to migrate. They're migrating out through the microholes and towards the growth factor gradient into the parenchymal space. And this is really the first step of angiogenesis. When that happens, the permeability goes up a lot. And these microholes we also used for the reverse process. When we grew cancer tissue around these structures, then we were able to see metastasis happening, where the cancer cells would actually move into the blood vessel lumen and go into the downstream compartment if the two tissues were connected. We can use an inflammatory factor such as TNF-alpha, and if we apply TNF-alpha to the media that's flowing through these channels, we're going to activate the endothelium, and then when we apply monocyte, they will be rolling and they will be extravasating into the tissue. So this just shows we can capture some of the expected processes, that this is how we expect the blood vessel network to work. And we would take, for example, cardiomyocytes from both rat and human embryonic stem cells. We can seed it around this vasculature, and we're going to get a tissue that's twitching, that's propagating electrical impulses. And here we can also apply drugs through the endothelium, and these drugs will then diffuse into the tissue space, and they will affect the beating of the cardiac tissue. So Boyang was very persistent. This was actually a reviewer question. They asked us, how thick can you make this tissue? He was very persistent, and he made this quite thick angiochip scaffold, a multi-layer. When you do that, you have to centrifuge the cells in. They're not, if you seed the cells, if you want to have uniform and high cell density, if you just pipette things on top, they're not going to sink in, right? You have to force them into the tissue by centrifuging. And then you have to grow them with higher pressure head. So he modified his bioreactor to provide enough flow. And then we were able to get a tissue that was quite thick, as thick as the left ventricular wall of the adult rat heart, and that it had cells uniform throughout the entire tissue. What's also neat about angiotrip is that it can be connected directly to the circulation. Usually, for this to happen, you will have to harvest the vascular bed from one part of the body and connect it directly as a flap to another part of the body. But with angiochip, we can just take it and literally insert it into a blood vessel. And here in a rodent model, it's a rat, we would, for example, cut the artery and we can insert the angiochip, establish the perfusion, or we can create an artery to vein graft. And you can see that when we let the blood flow, there is no thrombosis. Everything looks pretty well. Interestingly, you don't need endothelial cells for this to happen. The polymer itself is pretty good because it has citric acid, which probably partly acts to inhibit platelets because it chelates calcium. You can see that we can establish perfusion immediately, directly hook up the plumbing. And these blood vessels remain open like this for a period of 1 week, at least. The smallest vessel here is about 50 microns, 50 to 100 microns, and the inlet and outlet is 1 millimeter. So it's quite below that 6 millimeter size that we expect to be patent. So angiochip requires about 2 million cells, and 2 million cells is still a lot, especially if you're using iPS cells or human embryonic stem cell derived cardiomyocytes. So we scaled everything down to a size of 96 well plate. This is basically 1D angiochip, and we use only 100,000 cells per tissue. And the bottom of this plate is customized. I'm going to show you that in the next slide. And using this platform, which we call the INVADE platform, we were able to develop models of blood vessel, liver, heart, tumor, to use this system to study SARS-CoV-2-induced inflammation as well as pollution toxicity. So the bottom of this well is created by hot embossing in the craft facility that I mentioned. So you have a little well within a well, and into this we put our polymer blood vessel with these microholes. And in some cases, we can also embed electrodes if we want to stimulate. We've so far only connected 2 tissues, which is probably enough. As you connect more and more, it becomes actually quite complicated. You could connect, for example, heart chamber and liver chamber, or you could connect liver and cancer to study communication through the common vasculature. And you can see it's the same process. First, we endothelialize, then we seed parenchymal cells, and then we can look at contraction. And here you can see the case of really nicely endothelialized lumen. And this concept of pressure-driven, it's basically just gravity-driven flow, where you have more media in the inlet compared to the outlet, and the flow just goes in that direction. Because I have microholes, as you can see in this graph, endothelial cells are really controlling permeability of this structure. And without microholes, the permeability is actually quite low. And the cardiac compartment has cantilevers. These cantilevers are a bit tricky to produce, but it's possible. And then you can see as the tissue beats, cantilevers are deflecting, and we can use calibration curves, force displacement calibration curves. You can see that here to basically look at the force of contraction. And then this conduit here, the channel is endothelialized. And you can see cardiomyocytes. This is troponin T staining. They're spanning the cantilevers. What we are trying to do now is something like this, where you have microvasculature connecting to this larger 100 micrometer in diameter vessel to get more like hierarchical branching. So I'll tell you one story that I think is special. We just published on this in Science Advances. It's using this platform to study inflammation that results from SARS-CoV-2. And so this paper is very important to me. It's a very special paper out of all the papers that I published because my grandma, shown here, died of COVID-induced heart failure in the ICU. So she was immunized. She was also 89 after she got COVID. COVID went away, but she got heart failure and she unfortunately died. And there are quite a few patients who had the same experience. So what we did here, we built the heart tissue around this angio tube invade platform. And if you just have the heart tissue, you can see this is troponin T staining green. The polymer is autofluorescent blue, endothelial cells are inside. Everything looks pretty normal, and the tissue is beating. And this is like at different frequencies, we stimulate at different frequencies, so you see the rate is changing. If you add SARS-CoV-2, there is less cardiomyocytes, there is some cytotoxicity, but it's not too bad, the tissue is still beating. But now if you perfuse peripheral blood mononuclear cells, many of which are monocytes derived from the bone marrow, these are actually from real human blood. Now you see there is extravasation of these immune cells into the parenchymal space, and basically the heart flatlines. This is followed also by a very robust and profound secretion of inflammatory factors such as IL-6 and IL-8. And if you look at the TEM, at these different cardiomyocytes, the one thing that you see, okay, control looks pretty decent. You have some sarcomeres, you have some mitochondria there. You see a loss of sarcomeres, which is what you expect if you don't have contractility. But what was also very dramatic is that we have basically destruction of mitochondria. The mitochondria are almost gone. And you can see in this SARS-CoV-2 group plus PBMC, these large exploding mitochondria. And if you take the media, you can actually measure very large amount of free mitochondrial DNA in this supernatant. So we thought, okay, this is kind of interesting. We kind of stumbled upon this finding because we collaborated with Anna Andreasa, who is a really mitochondrial expert. But does this have any relevance to anything that's happening in the patients? So we reached out to Claudia dos Santos. She's an ICU doctor at St. Mike's Hospital in Toronto. And she has banked basically plasma and serum from the patients who were in the ICU, who she was taking care of. And she had this sample collection from before the vaccine was available. And so when we measured free mitochondrial DNA in these samples, these are the data that we obtained. If we look at COVID-negative ICU patients, free mitochondrial DNA is pretty low. If we look at COVID-positive ICU patients, and then we break them into two groups, those that have left ventricular ejection fraction higher than 50%. Those are patients who don't have affected cardiac function. And then we compare that to those who have ejection fraction under 50%. We see that patients with compromised contractile function who are COVID positive have huge, very, very high levels of free mitochondrial DNA. And interestingly, all the red dots are people who didn't survive. So there is a lot of people who didn't survive in this group. Then the question is, can we do something about this? And our hypothesis was that this is really triggered by inflammation. Free mitochondrial DNA is very highly inflammatory, can trigger inflammasome, TLR activation. We said, let's try to calm down, make sure that the endothelium is calmed down. So we used exosomes derived from very patent monolayer, happy monolayer of endothelial cells. And we use them as a therapeutic. So when we perfused exosomes through heart on a chip that was infected with SARS-CoV-2 and with immune cells, we see that in the presence of these exosomes, there is a lot less extravasation of the immune cells and a lot less secretion of pro-inflammatory factors such as IL-6 and IL-8, MCP-1. And as a result of that, there is a lot less secretion of free mitochondrial DNA. You can see that in this group treated with EVs, we do see some mitochondria at the same time point present. And ultimately, this enables us to rescue this cardiac function, at least in the organ-on-chip system, to make sure that these exosome-treated, infected, and immune cell perfused hearts on a chip actually beat quite well, and they continue to fire calcium transients. So this demonstrates how we could use a vascularized heart on a chip to discover a potential biomarker and test a potential biologic therapeutic. But I talked to you about fractality of vasculature, but I never showed you anything fractal. You just showed like one channel, or I showed you like a channel branching in X, Y, and Z direction. I never showed you anything that looks like this. And this is not a real picture of vasculature from the heart. This is also an AI-generated image that is just supposed to capture your imagination. So using microfabrication, we can create structures that are 1 millimeter, 500 micron, half a micron, and we can also create structures that are several millimeter. But to go down to like 5 micron, 10 micron diameter vessels, we cannot do that with microfabrication. We don't have the resolution. So we have to do it in a different way. So Shira Landau, when she came to my lab from Technion, from Shulamit Levenberg's lab, she wanted to work on microvascularization of the heart tissue. So she used this system consisting of endothelial cells like UVEX together with stromal cells. And the stromal cell that she uses is dental pulp stem cells. So this is a little bit artificial system. It's not isogenic, but it's good because we know that we can make very robust vasculature that persists quite a long time, that has good gene expression of matrix proteins as well as proangiogenic proteins. If we use combination of these two cell types, we get like microvessel formation. So one obvious thing is, well, let's just add cardiomyocyte to this combination. So now you have 3 cell types. When you do that, not much happens. So look at this bottom row here. When you add cardiomyocyte, this vasculature, kind of microvasculature, goes away. And you could say, well, you just have to work harder, right? Why don't you try different percentages of endothelial cells and different percentages of cardiomyocytes? And when we do that, you can see that the more cardiomyocytes you add, the less vasculature there is. And it's almost mutually opposing. If you have a lot of endothelial cells, you're going to get good vasculature, good perfusion, but no beating. And if you get beating, then there is no vasculature there. So working with Slava Eppelman and relying on these more recent single-cell sequencing studies, it became really apparent, in the heart, there are cardiomyocytes. We know that. And there is endothelial cells, there is fibroblasts, but there is a lot of immune cells. And one of those very important immune cells are resident macrophages. So Slava Eppelman is a cardiologist in Toronto General and my collaborator. And during his postdoc about 10, 15 years ago, he was one of the first people in the world to describe and characterize resident macrophages in mouse hearts. And they arise from the yolk sac, and they're very important for regenerative responses as well as angiogenesis. And then, but where are we going to get these cells from? So fortunately, we have Gordon Keller also at the Toronto General Research Institute. And also some 10 years ago, Gordon Keller published a protocol of how to differentiate hematopoietic stem cells from human embryonic stem cells. And the same protocols can be used for iPS cells. So using these protocols that are basically developed by Gordon, we were able to differentiate something that looks like a primitive macrophage. So these cells express LIVE1 and they're CCR2 negative. People usually also ask us, how about other markers that map with M2 macrophages? They express those markers as well. And when Shira took these macrophages in co-culture with cardiomyocytes, endothelial cells, and stromal cells, now this is 4 cell types, she was able to very nicely stabilize the vasculature. You can see endothelial cells in green and macrophages in red, and they persist in this 4 culture. It's really important that the macrophages are in the tissue. If we put them in the transwell, we don't get as much benefit. A little bit of benefit, but not as much as when they're really right there in close proximity. Here, we also use that BioWire as an assay just to figure out how do macrophages contribute to contractility. And we can see that despite this being non-intuitive, you're adding more non-myocytes, you're diluting cardiomyocytes. Actually, the tissues with macrophages beat quite well, better than the tissues without macrophages. And you can see they have really nicely elongated cardiomyocytes with sarcomeres. We actually, together with Slava led a paper that we just published in Nature Cardiovascular Research about a month ago that really looked at the system in a lot more detail in the mechanism of how macrophages are helping in such tissues. And because I think a movie is like a million words, right? They say a picture is 1,000 words, but a movie is like 10,000 words or a million words. If you look at contractility, right? This is just a fibrin dome, like with cardiomyocytes that you cannot see, macrophages in red, and endothelial cells in green. You can see how nicely the tissue is beating. And this is the contractility in equivalent tissue without macrophages. The movie is running, but there is really no contractility. If you look in the BioWire assay, again, you can see that this string of cardiac muscle is beating a lot more vigorously compared to with macrophages compared to the one without macrophages. And then the question is, do these cells really help with perfusion? And the answer is yes. With macrophages, we get nicely perfusible vasculature on par with what you would get in vivo. And you will be able to see that in this movie, in the movies here that are running. If you look on this side, you can see blue beads going through the vasculature. Here we are using iFlow plates from Organobiotech, which is Boyan's company. I don't have any equity in it. If we don't use macrophages, we just get like a sheet of cells. And if we use macrophages with cardiac organoids, EBs, we can get these endothelial cells to wrap around the EBs, and you can see blue beads passing through. So one of the big ways that macrophages help is they clean up the environment, right? They eat up all the free mitochondrial DNA and the cargo from the dying cells. Keep in mind, this is not a happy environment. You keep it starting from isolated cells, but they also have a direct effect on angiogenesis. When we did single cell RNA sequencing, we saw that there is quite a few populations there. There are also fibroblasts that arise from directed differentiation. And basically, in this 4 culture, macrophages are telling stromal cells and fibroblasts to become more pro-angiogenic and signaling through hepatocyte growth factor and insulin growth factor binding protein 7. So I showed you a few technologies from my lab where we showed that basically we are controlling structure to achieve better function. And I showed you a few tools like AngioChip, Invade, BioWire. We also saw that in plastic platform, we can do studies of SARS-CoV-2 as well as the macrophages help with contractility. I'd like to thank all the collaborators and our funding sources, people from our lab. And just here is a slide for Kraft. We are happy to collaborate. We have really very, very nice state-of-the-art microfab facility that's quite unique because it can process PDMS and plastic as well. If you haven't heard of this journal, ACS Biomaterials Science and Engineering, I'm an executive editor, and we're always looking for good biomaterials and microfab papers. So thank you very much for listening to my lecture, and I'll be happy to take your questions now.
Yeah, thank you very much for this very exciting presentation. And I'm sure there are many, many questions. Who wants to go first? Maybe, okay,
Mike, you will go first.
Yeah. Just because I sit in the front row. But I have a question with the macrophages. So these were iPSC-derived macrophages, and you mentioned they're sort of primitive macrophages.
Yeah.
Have you, it's sort of that question, what kind of macrophage do you need for this to actually work? Did you also try this with like primary monocytes? And would they sort of reprogram to become more tissue-like or?
Yeah, so a macrophage is a human embryonic stem cell derived. That's a standard protocol from Gordon. So the tissue is a little bit of a Frankenstein. it's not isogenic. And definitely through single cell in the paper that Slava led, and in this one, there is a change of the macrophage as they spend time in the tissue. From single cell, we know that they become more like cardiac resident macrophages the more time they spend in the tissue. With that being said, we haven't done the control where we use blood-derived monocytes. So maybe it's also possible to do it. If you polarize them towards M2, maybe you're going to get some beneficial effects of that cell as well.
There's actually one question in the chat. How far is it to mimic also the impact of the microbiome? I know that may contaminate your chips, but maybe it can be flooded with the components the microbiota produce.
Yeah, well, definitely that would be really important for if you're working with something like gut on a chip or skin on a chip, right? So I would say in most of the cardiac applications, we only applied one virus, right? We didn't go into complexity. The only one virus that we applied is SARS-CoV-2, or we would apply common cold coronaviruses like NL63 of 229E. So we didn't really apply anything more complex where you would have several of them. And I think that's definitely important for gut on a chip.
Yeah. Okay, a couple of questions in the audience.
Yeah.
Yes, thank you. Very, very nice presentation. I was wondering because you mentioned structure and function and also the size of these tissues. And I was wondering if you have kind of data that by having vasculature inside, then you can increase the size and keep the function, which I guess is something that we would love to have.
Yeah, so I'm not that enthusiastic right now that we can build something that's many centimeters in size, right? The largest that we have built is basically under 1 centimeter, right? It's basically just several millimeters in thickness. But for cardiomyocytes, and I like in my own hands during my PhD, they will die beyond 100 microns, like 200 microns tops, because the density is very high. You need high density. It's not cartilage. You need high density for them to have good contractile force. They need to be connected by gap junctions. And if you go beyond 1 to 200 microns, everything is dead. I was trying to make these like big tissues, even by forcing interstitial flow through a scaffold, you get M&M candy. Like on the inside where it's chocolate and M&M, it's all dead. On the outside, you get living cells, right? So with these approaches that I showed you with angiochip, we can make this 1 millimeter plus, right? But I'm not optimistic that we can now make the whole like big sort of human-sized heart tissue with this yet. But we are going I think we're going in the right direction. We have all the ingredients in terms of, stabilizing the microvasculature, doing the branching. I think vascularization is a perfect problem. You can spend your entire career working on it.
Yeah, fascinating work. Thanks a lot for the talk.
Yeah.
I was wondering, how would you categorize your cells or the cultures in terms of fetal to postnatal and adult stages? Are they more fetal character or more adult character? And can you increase this or push it towards adult character by having more components in there? Like
we had so this last part of the work on vascularization and even the SARS-CoV-2, there was no electrical stimulation. We didn't stimulate. And with macrophages, we didn't stimulate on purpose because we didn't know what would be the effect of electrical stimulation on macrophage. We started stimulating some tissue and we saw that the shape is changing. There is Michael Monaghan. He is at Trinity College Dublin. He actually has similar iPS-derived macrophages. He's found a protocol, electrical stimulation, that they like, right? You don't want to polarize them towards something that's pro-inflammatory by stimulating. So we didn't stimulate, but we had the papers before where we just focused on maturation. And by stimulating with electrical stimulation, you can push them towards more mature phenotype. Is this adult phenotype? I would say probably not fully adult. Well, is it fetal? It's probably kind of past fetal, right? It's somewhere in between. What's also interesting, it depends on what you look at, right? During development, most likely things develop perfectly together like they follow one another perfectly, right? Sarcomeres and ion channels. And so we saw with this application, we push ion channel maturation ahead of the sarcomere. So it's a little bit artificial system. Gordana had a nice paper in Nature where they used the post system and they got a structure that's almost indistinguishable from native heart structure. So keep in mind that in these in vitro systems, we don't push them at the same time perfectly like in vivo. And then there are also metabolic maturation protocols that we didn't superimpose on this, right? So that you can push metabolism further.
Hi, I'm Pragati from the Gotthard lab here. So we work quite a bit with the Eschenhagen model of engineered heart tissues.
Yeah.
And I was wondering if you've looked at this larger cardiac patches developed by the Weinberger group, where they kind of have perfusible, probably, but a larger patch of engineered heart tissues, where probably I think if you integrate that in this approach, you could actually get vessels to sprout into the EHT, I suppose.
Yeah, so we made patches, like injectable patches, pretty large. I have them in the slide. I'm not sure if I'll be able to go back, but it's more for with the view of transplantation and doing kind of layer by layer. Oh, I can't project it anymore. Layer by layer approach. And I'm aware actually of Zimmerman's. He's doing a clinical trial by placing the patches on top of the heart. So we have, using POMAC polymers, we were able to make injectable patches that you can inject through keyhole and they open up at the site. And they beat actually quite nicely. And then the idea is you could have several thin patches and just keep layering them on top of one another. The problem with the patch approach, it's uncertain if there will be ever integration of these patches because the epicardium is pretty actually thick. And if you look at the rodent epicardium, is thin and you don't think it's a big deal. But then if you look at, let's say, porcine or human epicardium, you can see there is a lot of cells there. And they really form like a barrier to integration.
My second question was also, for example, with the IYO with the macrophages, do you see any accelerated remodeling of the tissue post casting? Because I think when you had multiple cell types, there's also like remodeling.
There hasn't been really that much of a difference in the way the tissues were compacting, and the final size was actually quite similar. So at this percentage that we were using, there hasn't been a lot of difference in the size or the rate at which they were remodeling. It was quite comparable.
Thank you.
There's actually one more question in the chat, not that one, but the other one was about if the combination between cardiomyocytes and macrophages helps with the electrical coupling.
Oh, yeah. Yeah, so that was a paper in Cell, right? They found that macrophages can couple to cardiomyocytes. In this paper, we haven't directly looked at coupling in a rigorous way. You would have to inject a dye and see if it goes through. But we see by bulk RNA sequencing, increase in connexins. We see that the group with macrophages has upregulated the connexins expression, which would suggest there is more coupling. I just don't know if there is direct coupling between macrophage and cardiomyocytes.
Okay. And for the sake of time, I'm reading now the final question, and there will be ample opportunity for discussions in the PhD and postdoc lunch now, and then also throughout the next 3 years. So the final question comes from the virtual attendees, and Mario asks, could you mimic aging and study aging effects by making structures with young and old senescent cells?
Yes. So I've seen some work. It hasn't been published. I've seen as an examiner a really nice work where they would derive cells from progeria patients and see if they would accelerate aging. And I think that's a really great use of these organ on a chip systems. My only caution is you have to figure out if your cells are matured properly, right? Do they go through this like fetal, healthy adult and then aging? Or is it just, oh, they're poorly mature, so that's why they, like the disease phenotype and aging phenotype in cardiac often turns on the fetal program. So you want to make sure that you matured your cells first before you conclude that they are aging, because they could just have that phenotype because they're fetal or immature in some of the properties.
Okay, so let's thank Milica again for this fascinating and inspiring talk. And thank you all for coming and joining today. Thank you.